So actually I was told to make a version in English. So this is an English version. So we are going to study how HIV enters into the human body cells. So we are starting with by drawing the structure of the virus HIV. So we are drawing the structure of HIV. So let us first draw the structure of HIV. Yeah, we are done with the reverse transcriptase enzyme. We are done with the RNA. We are done with the covering of the RNA, which is the nucleocapsid. We have also put inside there the protease enzyme. We have also put there the integrase enzyme. So that is our enzyme. That is our HIV cell. That is our HIV cell. So this cell all over it has proteins P120 and P41. So that is P120. It is all over it. See, so it is surrounding all over the HIV virus. It has those. We could call it legs, or it is used as its legs or its hands, which helps it to attach to a host cell. So the top of it is the P120, then what joins it to the virus is P120. So we have this part, it's called matrix core. So the next part is that is the capsid. That part there, it is called the capsid. Then that part covering the RNA, that one covering the RNA, it is called the nucleocapsid. Then you can call it P7, that one P24, and that one you can call it P17, because those are proteins. They are named protein 7, type 7, like that. Then inside the nucleocapsid we have the RNA, or the ribonucleic acid. Then down there we have the enzyme. That enzyme is called reverse transcriptase. So we are remaining with the two enzymes. This enzyme is called protease, and that remaining enzyme is called the integrase. So that is our virus. So now we want to say how it, when it enters the body cells, how does it again enter into the human cell, the host cell? Because here there are two things. There is getting it like from the major source, the sexual intercourse. Now, once it enters into the bloodstream, how does it enter a blood cell? Because in the blood we have different types of blood cells. So one of them is where it enters the T helper cells. Now the T helper cells, they have a molecule. So let us draw it here and see. But as if we have drawn a smaller one, let us draw a big one. Let's draw a bigger one. Let's draw a bigger one. Yeah, this is our T helper cell. But so our T helper cell here, this big one, yeah, the big one we have drawn, it has a molecule which we call the CD4. That's why most of the people talk about CD4 cells. CD4 cells. They are called the CD4 cells because of this molecule. It has that molecule called CD4. CD4, it means cluster of differentiator 4. So since our since this okay it is our since our HIV cell has those the we in a lay language, let's call them the hands, but they are not really hands, the ones which the one protruding all over the HIV cell, it has them. So it looks for where to attach, but it looks for somebody who also has the same, which looks like has, which has the whereby there is the possibility of it attaching to those things. So even this cell has things which can allow the attaching, so it attaches there. Yeah, the HIV cell, the HIV P120 comes and attaches on the CD4 molecule, which is on the T helper cell. Understand? Yeah, they attach together. So after attaching together, there is also another thing on the P120. Another one, which is P41. It once this one once P120 attaches on the CD4 cell, they pull one another. The P41 also comes and attach into the cell, into the cell wall, and later they come together, and this, yeah, the cell and the cell and the HIV cell, they become together, eventually entering into the human cell, the T helper cell. So once they enter into the T helper cell, they are there like that. The other outer coating is left outside. The outer coating is left outside. The envelope is left outside. What only enters the body is the content inside that outer outer coating. So we only have now in the cell the RNA and its coating of the RNA, which is the nucleocapsid. Now, once they are in the cell, So now it is in the cytoplasm of the T helper cell. They become uncoordinated, and that one, the outer, the capsid, once it is uncoordinated, it goes away. Even after some time, another coating called the the uh, the capsid goes away. At first, it is the nuclear core which goes away. I mean the matrix core which goes away. Next is the nuclear capsid to go away. I mean the capsid to go away. Now here in the cytoplasm, we also have a nucleus aside. So the nucleocapsid covering the RNA has also gone away. Now we have remained with only the RNA. We have now only remained with the RNA with our three enzymes, the reverse transcriptase, the protease, and the integrase. And our RNA, those are the only things we now remaining with in the cytoplasm. So now, the reverse transcriptase now starts to move along the RNA, and once it moves along the RNA, it changes it from the RNA state to a DNA state, and we now have a DNA. So it again moves along that one DNA and make it two DNA. It makes it two DNA. Now once it has changed to two DNA, now we have two double-stranded DNAs. So they are now double-stranded DNAs. So once they have been made like that, they are now fully double-stranded DNAs. Remember, we have our nucleus aside here. In that cytoplasm, we have a nucleus which is here. So in this nucleus, it has its own DNA. We have a DNA inside here. So the DNA of the virus enters into the nucleus and moves away from the cytoplasm. It enters into the nucleus where there are DNAs. You see, it enters into the nucleus where there is the DNA of the. So the integrase also come and enter the nucleus, and cuts there and cuts there, and joins the DNAs of the virus to the DNA of the human cell, and it becomes one DNA now joined together. And once this happens, the cell has become infected now for life. And actually, even if you go to church, even if you go to the masjid, even if you go to the shrine, there is no reverse now. Once it happens, it has happened. So now we have our DNA inside here. It is this DNA which is inside here that is used to produce more RNAs. It produces various copies of RNA, and they produce a lot of messenger RNA which come into the cytoplasm. And it is these messenger RNAs which help to produce more other proteins, which proteins help to form another virus or other viruses. So they become so many RNAs in the cytoplasm being produced from the nucleus. Are you seeing how it becomes many without even its own production? So in the cytoplasm, we have our protease. This protease keeps on cutting these RNAs into small pieces. Hence, they come together and form and get a cell wall, and once they come together, they start forming another virus, which also move out, and the coating comes back onto it, and once it gets that coating, it has now become the new virus that can infect other cells and do the same process as it has been doing. And when you next time play unprotected sex with an infected person, you are ready to infect that person. So that is it. That's how it is infected. That's how it infects a human soul. So don't forget to subscribe. Don't forget to like. Don't forget to comment your question or how you have understood, or where you have not understood very well. Or actually, I'm going to bring much. We have chemistry is coming. We have actually we are going to the next part to tell how it can how it affects. But actually, slightly we did not talk about here the I forgot to tell you something small here about there are some other components of the cell here. We have the these ones. They are called the CCR5, actually OCXCR4. This is CCR5. This one's once the CD4 is in touch with the P120, it also comes and confirms the attachment. Just keep it from family attached. So that's what we had not talked about, but much we talked about it. So you can subscribe for more information. Actually, comment the topic you want us to talk about. Subscribe to Dr. PCM. That's my number. So thank you for watching. Thank you for everything you've done for us. So have a good stay.